Good morning to everyone, and thank you for joining this seminar today. It's a pleasure to introduce today the speaker, Dr. Jelle Penders, who is co-founder and CEO of SPARTA Biodiscovery, which is a spin-out from the Imperial College London. So Jelle completed his PhD at Imperial College in the group of Professor Molly Stevens, where he specialized in novel methods for nanoparticle and nanomaterial characterization using Raman spectroscopy and electron microscopy. And later, during his postdoctoral work, he co-invented the SPARTA technology that is an advanced technology, as you will see in this presentation, for the characterization of complex nanoformulations. So thank you very much, Jelle, for accepting our invitation and being here. And please, when you can go ahead.
Great. Thank you so much, Elizabeth. So it's a pleasure to be here today. Thank you so much for the invitation. It's lovely to visit Barcelona and, of course, to talk to you here today. Today I will talk you through what SPARTA is, the background, and why such a technology we believe is very necessary for all the future work that we believe needs to be happening in nanoformulations to develop the best, most powerful formulations, but also the best quality of formulations from a safety perspective, of course, as well. Just a brief introduction. Elizabeth has already done that. Let me see if this thing works. So a really brief background. So I'm originally from the Netherlands. Did my undergrads there, chemistry, chemical engineering, and then moved to Sweden for a bit of work, mostly on nanotechnologies, electron microscopy. And then indeed did my PhD with Molly Stevens at Imperial College, which really was a wonderful experience in a Marie Curie PhD consortia, where we started to lay the foundation of what nowadays is SPARTA, so single particle automated Raman trapping analysis. I will talk to you to the concepts of the technology, and then we founded the business about 4 years ago now in March. It was 4 years, which is based in Scalespace in White City in West London, which is a really cool spin-out environment where a lot of startups are to grow in that space. And pleased to say that this year we're expanding quite a bit. So we're going to be about 15 to 20 people now in SPARTA. As a company history, indeed, so the original work was done in the Stevens Group around 2016, between 2016 and 2020, where we filed the first patent and the publications, and we founded the company, and we funded that, and we work a lot with our core partner, Sartorius, you might know, is a big German-based multinational that is helping us basically bring this technology to the masses, especially from an industry perspective. The core instrument was launched in April 24, the Aegis 1. So what is SPARTA and what do we actually work with? So with SPARTA, we look at many different kinds of nanoparticles. So it's a nanoparticle analysis technique. And contrary to a lot of techniques you might be very familiar with, like dynamic light scattering or nanoparticle tracking analysis, things like that, we don't just look at size, we look really at the composition of these particles. So we use a technique called Raman spectroscopy, which we'll explain in a bit more detail, to look at the chemical variation within these particles and on a single particle level. Now, what are some of the particles that we look at? We can look at lots of different things. Roughly, for SPARTA, we look at particles around 40 to 400 nanometers. In some cases, we can go a little bit smaller. There's some cases we can go a little bit bigger, but that's the broad range of particles that we look at, which encompasses almost any kind of nanoformulation that you can think of. So, of course, we work a lot with liposomes, LNPs. I'm sure you're all very familiar. I'm sure that a lot of you already have them in the body from the COVID pandemic. So the Moderna vaccines or the BioNTech vaccines, for instance, which are lipid nanoparticle based. So very exciting advent here in medicine, where we have these nano-based carriers of different drugs or nucleic acids that are able to cure different types of diseases from a vaccine perspective. But there's lots going on beyond COVID of vaccines in terms of cancer therapies, for instance, or gene therapies that are based on LNPs. So there's a very bright future ahead there. We work with viruses as well. Chiefly here, it's adenoviruses or lentiviruses. So again, for lots of different types of therapies, people want to investigate these particles further. There's a very wide range of polymeric nanoparticles. So sometimes they're called polymersomes as well. There's also people that incorporate these more bottom-up design chemistry for different nanoparticles and different carriers. And we work quite a lot with exosomes or extracellular vesicles as well. You might have heard of this. These are these cell-derived nanoparticles. So basically, any cell in your body produces these tiny nanoparticles that are called exosomes or extracellular vesicles. And they are liposomal-like structures that are used for cell signaling and also implicated in different disease states, especially in cancer. So we have all these very exciting nanoparticles, lots of research being done, lots of products getting towards the clinic on this, but we need to analyze them to know what we've made. And what we saw was a big gap here in the types of the analysis that could be provided. So there's lots of techniques that can look at the size of these particles, but almost nothing that can really look at the composition of these particles, apart from using very established techniques like HPLC-MS and so on, where you break up all these components and then look at the slurry. And whilst that gives you, of course, the composition that the bulk of these particles are made of, we're losing a lot of information here that's very relevant, namely the variation between these particles in terms of the compositional profile. So because all these particles, how hard you might try with your formulation, are not created equally. There's a lot of variability there, and that's exactly what we want to measure. So why is that important? So say you have these kind of particles, there can be ligands inside, there can be drugs inside. We've got a complex membrane. But what you're actually making probably looks more a little bit like this. So we've got particles that are a bit bigger, some are a bit smaller, some have lots of certain components, some have less of certain components. And if we then do a bulk-based analysis, it will just get one data point, and we might be able to compare different batches of formulations, but not look within that batch. And what we say is that basically for every kind of component in your particle, there is a distribution here. And we can look at different vesicle components or cargo or surface moieties, and they all might have different distributions as well. So it doesn't mean that there's only one type of distribution inside your formulation. It could be that it's very homogeneous in the amount of ionizable lipid in your LNP, but very inhomogeneous in terms of the targeting ligands. So this is really where we need much more advanced chemical analysis techniques that can actually profile all the different components and what you're actually interested in your type of formulation. So what can we do with SPARTA? So SPARTA is a Raman spectroscopy-based technique, which I will explain what that means. But crucially here, we combine that with optical trapping, which we use a laser to look at one particle at a time. And that allows us to get single particle resolution. So again, as I've just been explaining, this gets away from the whole bulk-based aspect of analysis to really look at individual particles, how much variation there is between these particles. We want to use a label-free technique ideally. So with SPARTA, you only need to dilute your sample. We don't need to add anything to get the standard signal out of the particles. So that's really important. And we also look at the native state of particles in solution. So just your vial with your particle, whatever the particle might be, we can put under the machine in liquid state. So we don't have to dry the sample. We don't have to adhere it to a surface or things like that. We can just look in the native state at the particles directly. And we can look at many different things at once. As you can see from this Raman data, it's very rich. And we can look at stability, we can look at targeting, we can look at loading, all these things at the same time, which makes it very economical that you don't need to use a different machine for every single use. Very broad range of compatibility in terms of types of particles, but also in terms of buffers or things like that we might be working with. And we try and make it also easy to use. So it's specifically designed to be used for formulation scientists that might not have used Raman spectroscopy before or might not be very familiar with these more advanced techniques. Especially from taking a measurement perspective, we make that super, super easy. Okay, great. So what does SPARTA actually based on just from physical principles? So as I said, SPARTA stands for single particle automated Raman trapping analysis. And basically what we do here is we combine Raman spectroscopy with laser optical trapping and a whole bunch of proprietary algorithms to make this all easy and fast and automated. And I'll explain these techniques in a little bit more detail. So Raman spectroscopy, you might have heard of. It's a spectroscopy technique that was developed, I think, even over 100 years ago, but really seen a big boom now, especially because for Raman spectroscopy, what's essential is very strong monochromatic light, i.e. lasers. So now the lasers are getting much, much better all the time, so we can get a much better Raman quality signal. And we see how Raman was developed by a Nobel Prize winner, and it was used as a very niche application first, as a very weak effect. But with these better lasers and the advent of different applications from material science all the way now into actually life science, it's getting a lot more adoption. The most prevalent kind of Raman scattering that you might even have come across without knowing is at airports. They have scanners sometimes for water bottles or things like that. That's all based on Raman spectroscopy. That will tell you if there's anything else in the solution other than water, for instance, that you're allowed to bring in. So it's used very heavily in detection techniques, for instance, also for olive oil. As adulteration of olive oil is a big problem, that people mix in cheaper oils with the olive oil. So you can use Raman spectroscopy, for instance, as well to see that. So these are all like very practical niche applications. There's a lot going on in biology and in life science as well. Before I explain more the physical concept of it, there's also different kinds of Raman spectroscopy that people use that you might come across in papers as well. So we use something called spontaneous Stokes Raman scattering, which is kind of the neutral flavor of Raman spectroscopy. There's also something called SERS, so surface enhanced Raman spectroscopy you might have seen, that has I'm not going to explain that exactly how that works, but basically there's a gold interaction with a nano-based surface to do some sort of signal enhancement. There's CARS, which is an imaging technique for anti-Stokes Raman scattering. So there's lots of different applications there, but we use the neutral variety here because it preserves the inherent signal variation in your particle the best, without causing specific enhancements, so that we can actually really look at the ratio between these chemical components. So contrary to other kind of spectroscopy techniques like UV-Vis or IR, we use one laser wavelength to interact with the molecules. So we use a near-infrared laser specifically because it doesn't damage the particles as well. So you could use a green laser, would get more signal, but it would also damage the particles at the same time, and we don't want that. As well as that, there's quite a lot of natural fluorescence in biological particles, especially when we're looking at cells or exosomes, which would interfere with the Raman effect. Now, what's actually happening? We shoot this laser at these nanoparticles, and all these photons hit the nanoparticles, and most of the light, the vast majority, scatters elastically with the particle, which means that the photon with one wavelength that comes in from the laser leaves the particle as well with the exact same wavelength. Now, this is interesting. You can do some scattering for size, for instance, of the particles of that, but you can't see any chemical information there. Now, a very small fraction of that light also scatters inelastically with the molecular bonds. And this is really the Raman spectrum, Raman effects that we're looking at. So what's happening is that this laser photon hits the molecular bond and then transfers a small amount of its energy to the molecular bond. And it leaves the particle with slightly less energy. And the amount of energy that it has lost is indicative of which molecular bond it interacted with. So we can then get out is a spectrum here. We use a very sensitive spectrograph and a camera for Raman spectroscopy to measure all these different, see if this works. Yeah, so you can see basically here at the intercept, it would be 0, which would be the light that didn't lose any energy, i.e. the same wavelength of the laser. That's not interesting. We block that light. And then all this light basically that comes off from these different peaks is basically more and more energy lost from the photon, so different molecular bonds that interacted with. And it's very well characterized at which positions here certain molecular bonds are interacting with the light, which means then that we can get the compositional profile or a compositional fingerprint of the nanoparticle that we're looking at. Now, that in itself doesn't allow us to do exactly what SPARTA is, of course, but it's basically where the signal comes from, which is an important thing to know, of course, when you're using a new analytical technology. So we combine this with optical trapping. So this is another Nobel Prize winning technique. It was about 2018, I believe, that Arthur Ashkin got the Nobel Prize for his advances in laser optical trapping. And this is a technique, you might have also heard it called laser optical tweezers, which is a variation of this. Is basically when you use a light focus with a laser in such a way that you get a very tight focal point, you can actually cause a physical force, very weak physical force, but a physical force on a particle or molecule that is inside that optical trap. So basically what we do is we have all these particles diffusing around with Brownian motion, and we have this very tight laser spot. And if a particle gets close enough, it stops the Brownian motion in that focal point of the particle. So we can have that particle kept still inside that point. And at that exact point where we have that laser focus, we generate the most Raman signal because it's the focus point of the laser. And that's where we can collect then the light off from that particle. We make this very confocal as well. So that means that we just get the light from the cross-section where your particle is and not from the light that is everywhere else, which means then that we get Raman spectral signature of one particle exactly in the focus point at a time. So that's where we combine that with optical trapping. And then the whole automation and the proprietary algorithms come in from SPARTA to make this all completely automated. So what was done before is you can trap one particle and have a Raman signal of that. Whilst that's interesting, it's not an analytical technique really yet, right? Because you have your formulation with your LNPs and you want to know what is the variation between all these particles. So we've made this completely automated, this whole complex process, which means that you can put your solution inside the machine, set a few parameters, press go, and it does all the interfacing and it does all the optical trapping, capturing data generation of single particles, all in a completely automated way. You can just walk away and then come back an hour later or so, and there's all your spectra from all your particles. So we make that part very easy. So I hope that explains a little bit how it works. You don't need to necessarily know that to use the machine, but we are scientists here, so I'm sure you appreciate to know the background of where the particles signal is coming from. Now, to just move into some examples of what you can actually do with this technique, and it will hopefully make it a little bit clear why this is so powerful to use for nanoformulations. We've worked with a lot of different institutes here. And I'm going to show you a wide range of applications. There's a lot of different things you can do with SPARTA. And I just wanted to show some people that we've worked with because I will be showing some of the data here. So what we do with SPARTA is really interact with the whole life cycle of this drug formulation product. So if we develop these particles here, for instance, in an institute, we are mostly looking at very novel components. We might have novel formulations that are, LNPs are already into industry. Maybe we're doing something really cool new that hasn't made it to the industry yet. So we need to prove here that what we think we made, actually we made. And this is where techniques that have a lot higher resolution can really help you prove that. But when we go take these formulations further, we might get, for instance, challenges in scaling up of formulations. So you're making this with a pipette or with a very small scale reaction in a lab. But in order for it to be a successful formulation in industry, we need to make this on liter scale, right? Or tens of liter scale, these formulations. And we use completely different reactors or different techniques to make that. And this can, again, cause different, variation in the composition. So we work with companies, for instance, that are scaling up this production to then test, okay, how do these different production methods actually interfere with the variation, and do I have the same formulation still that I've made? So you can do that all the way towards a quality control perspective, where people are actually making these formulations with a standardized process and want to check are these particles still the same, the ones that I made last week or 2 months ago or a year ago. So there's lots of different things we can do. And a key part here as well is that we want to optimize the use of resources. So a lot of times here, what happens with nanoformulation development is that people have limited techniques. So they make the particles, do DLS, do things like that. Okay, I've got the right size. We might be able to do a fluorescent assay to see if a cargo or something else is in the particles, and then we move into cells immediately. And whilst that's very powerful, it's very costly to do all these cell studies, and it's very time consuming. And usually from those cell points, we get only a few data points out if it's high or low or a percentage. We don't really know what has happened here. And what's very frustrating when we hear from a lot of people is that the formulation that I made last week worked really well, and then the one I made today doesn't work very well, and I don't know why. And this is really where this heavier investigation can really prevent you from doing a lot of work that is not going to be necessary, because maybe within our analysis on SPARTA, you could have told that formulation which you made today, there was a mistake in it, and we could have made it again, rather than waiting 2 weeks for my cell study to be done. So there's a lot of benefit here in this deeper investigation in an earlier stage of the process. Okay, so we get here a Raman spectrum. So this is based on lipid nanoparticles. This is a bit of a complicated nanoparticle. There's lots of things going on, like targeting proteins and things like that. But we get a spectrum out with all these different peaks. And we don't have to be very scared of all these peaks, because in our software, you can drop down these colored bars, basically, that will mark different marker peaks for the Raman spectroscopy. So it's not like NMR or like HPLC, where you really have to look at every little squiggle and see what is this, what is this, what is this. You can't do that as an academic exercise. It's fun if you like that, but for day-to-day use, we don't have to do that, because, for instance, for cholesterol, we look at the peak at 700, which is very characteristic for cholesterol, and we can look at the variation in that peak to look at the variation in cholesterol content. So it's really looking at specific marker signals that we then analyze in a univariate analysis way. So we go basically away from this Raman spectrum and really look at specific variations of different components. So there's a database in the software that helps you drop that in. And then I'll just talk to a bunch of different applications of other uses here. And you will see a lot of these kind of plots on the right, which is univariate analysis. So basically, from these 2,000 data points that we get per particle, we narrow it down basically to one specific peak or one specific peak area, when we investigate different components within a particle. And then what the beauty of that is that we can plot these univariate plots where every dot here is still a single particle that you've measured. So you have visibility on where are my particles basically for the distributions, but we don't have to look at the spectra all the time. We can investigate individual components as once, because they are in principle independent in the Raman analysis. So then we look here at the targeting ligands as an example. So this is, of course, a big field that's coming up. And what people are doing is they have a similar core composition and putting in different ligands on the surface. So we worked here with a company, and here we have the TP0, which is where they had 0 amount of targeting ligand, and we measure that signal. So it's very important with SPARTA that we measure a controlled sample that, for instance, doesn't have a certain component or doesn't have the targeting ligand as a baseline, because that baseline might not necessarily be absolutely 0. And there will be a bit of noise variation there. So then we add a small amount of targeting ligand, and we can see that formulation go up. And maybe about 80% or so of the particles here had the targeting ligand, and you can draw a line in the software to actually quantify that. And then they were adding more and more targeting ligand. And I just want you to imagine if you just got the mean values here of this, and no error or no other part of the graph, because that is what would a bulk technique give you, which is give you the mean value. It would give you a standard deviation, but the standard deviation has nothing to do with the variation within your particles. It has something to do with the measurement variation of how accurate your HPLC is to reproduce the same value, rather than saying anything about how variable these particles are. So this is really the hidden heterogeneity that SPARTA can provide, is actually showing like, okay, do I have a very narrow distribution or do I have a very wide distribution? And if we have such a wide distribution with particles that maybe contain 2 or 3 times as much targeting ligand as others in the same formulation, that's very important information to know how effective your formulation is, how good your production process is. And if you know that it's variable, you can start tuning things in your production process to try and make it less variable. I don't know what happened with this. Shall we just continue and see what happens? The lights went off a bit. Going in sleep mode? What do we think? What's it? Maybe wiggle the mouse. Okay, so when we saw this variation here, this is really where the company was then, okay, let's try and optimize this further. So I think the projector is not having a great day. Hopefully, we'll keep going. So what we can see here is then we have this native formulation without targeting ligand. And then with process one, this was kind of the first state of the formulation. So we can see pretty decent amount of particles have the targeting ligand on, but it's very, very variable. And then when they optimize the process, you can actually bring that variation down massively and have a very tight control over the amount of targeting ligands inside a formulation. And SPARTA can really help you show that that's the case, of course, and the difference there. Again, between process 1 and 2, marginal difference on a mean variation that you would have with a bulk technique, but of course, massive different insight on a single particle level. And a secondary measurement that we just do at the same time, you don't have to change anything in the settings or so, is based on looking at relative particle size of these particles. So on-target formulation was very nice. We see a relatively tight population, couple particles a little bit bigger. But when they had a lot of targeting ligand in that process one, they saw that it actually caused aggregation as well. And that's pretty common when people are adding that. But the nice thing is with SPARTA, we can then see that at the same time, but also look at some of these aggregates and investigate their spectrum and see like, okay, did they actually have, they just aggregated, but they have relatively small amount of targeting ligand, or is these also the particles that have maybe 3 or 4 times as much targeting ligand? So we can really investigate like, why are these particles aggregating? Or maybe it's aggregates from your particle ligand, from your targeting ligand. If you're putting an antibody on the surface in the wrong buffer conditions, it could easily cause aggregations of the protein, and you have free protein aggregates in the solution. If you put this on a DLS, it will tell you that it's aggregates. It won't tell you what these aggregates are. Again, this gives just a lot of insights for optimization. Then we're looking at some other particles. So these are mesoporous silica particles. And we're looking at doing different surface additions to these particles. So we can get a base nanoparticle spectrum here. And then we're adding different things on just so you can see that how that spectrum is actually built up. So we're having the bare nanoparticles, and you can see different marker peaks that we can draw down here. And then we're adding a polymer on the surface. And you can see we still have that same core composition, but a bunch of extra peaks come up here from the polymer that we coated on the surface of these particles. And again, we could look at the variation in that polymer coating as well. This was super homogeneous, which you can see from this average plus the standard deviation is this little shaded area. Again, this is the standard deviation that comes from the variation within your population of particles, not just from repeat measurements of the same set. And then we were adding here a nucleic acid as well on the surface. So you can see here, again, different peaks here that were added clearly into this formulation, and we can look at the variability there. And I think this was quite a good example to really see how a spectrum is built up. And quite important to have these controlled samples there as well to be able to identify the different signals. It's not 100% necessary. We have a database. It's very widely spread used. And even with Raman imaging techniques, for instance, that are not SPARTA, this Raman signal where the positions of where these peaks are, for instance, for nucleic acid, will be the same because it's calibrated. So we always know that these peaks are going to be there around 780 or so is a key peak for nucleic acid. So that's not going to change, but it's much nicer to see the control, of course, with the absence and the presence of the sample. And we can also use that to look at some loading analysis. So for instance, here we can see that we have a different ratio of particles showing nucleic acid signal and a different amount of nucleic acid spread in types of these particles. So this is all stuff that we can analyze. And we can actually see that the amount of loading, is you can calculate this in the software, is different on an average perspective. There's also other types of plots that we can make, so not just univariate plots. This is, for instance, a bivariate plot where we plot basically RNA signal against polymer signal. You could do this, also imagine this for lipids, for instance. And we can see really where those populations lie. And all these individual dots are individual particles in your formulation. So you really have visibility about how variable my particles are. Then another key thing, especially when we move towards more industry applications, is different contaminants in the sample. And this was a study that we did with Hansa Biomed on extracellular vesicles. And they had different isolations, basically, of the extracellular vesicles to use in different processes. And what we can see here is that there was a population of the product, there was actually two different kinds of contaminants. There was a protein contaminant and a lipid contaminant in there, which was mainly lipoprotein particles, because these were blood-based isolates of samples. And we can see this distinguishment purely on a label-free Raman spectrum. We can see the compositional profiles of these particles being very different. And we can quantify the amounts, basically the percentage of particles that have different compositions. And this is a really big problem in other kinds of production as well, especially when you get to larger scale production. There's also larger scale purification technologies, which usually have a lot of flow-based techniques. And what has become recently clear as a problem, that there can be leachables from all this tubing coming into your particles, which is contrary because it's a purification technique, so you're trying to remove things. Of course, it's removing, for instance, excess protein in the solution, but it can introduce contaminants as well. So the nice thing is we were able to detect this with SPARTA. We didn't know beforehand that there would be contaminants. So this is a big advantage of having a holistic technique like SPARTA, because we can investigate things that we're not necessarily looking for. If you compare this with a fluorescent-based techniques, no one is going to preemptively stain for silicone leachables in a formulation, or no one is really fluorescently staining cholesterol in LNPs, whilst we can see that there's significant variation can be between those particles. We can look at it all at the same time. So we can quantify the amount of this, and we can also track this through different process steps, and they use the different tertiary cleanup procedure basically to get rid of these contaminants. Critically, we could identify what the contaminant was and that it was associated with the particles as well, because we can really see it on the particle that we trapped. We saw the normal signal plus the contaminant signal, which again informs you what kind of purification method you might need to use, versus that it's a solution-based contaminant. And we can also compare this, for instance, to GC for, we did this for ethanol. So LNPs are usually made in ethanol part or other kind of solvents. And Raman is actually very sensitive in detecting organic solvents as well in the solution. And we can make a calibration curve here, and we compare that against GC actually on how accurate it was. And this is, again, a very easy test. Again, when you do all this normal SPARTA measurements, you get this basically for free. We can just look at a specific position. Is there ethanol left in your LNPs or not? And it's very sensitive to detect that. Cool. Now we've got some really exciting newer studies that were done on hybrid particles. So what we like to see is really in this field of nanoparticles, there's always trends, right? There's always hypes, and they are there for a reason. And I think there's two big things that are happening in nanoformulations. So people are moving towards a lot of targeting ligands because people want to do extrahepatic delivery. So the nice thing about the COVID vaccines are like they all go to the liver, which is where they wanted them to be for the vaccines. But that makes it actually really difficult for targeting other diseases. And you can also, if you remember the Onpattro, I believe, other formulations, hugely successful formulation, but it was specifically for a liver disease. And I think it's not necessarily because they wanted to cure that liver disease, is because everything goes to the liver. So let's target the liver disease because that's where all the particles are going. But we want to go to other parts of the body. So this is where targeting ligand comes in, which I've shown you a bit of data about. The other part, which is very interesting, and I think it's slowly moving out of academic territory to more companies, is hybrid nanoparticles. So this is usually tends to be a combination of polymer or lipid nanoparticles with exosomes, because exosomes have all these natural proteins and natural targeting ligands basically on the surface that we want to do. Pure exosomes, there's a lot of challenges as well in terms of production and control, and actually cargo loading, which is a lot easier with polymers or LNPs. But what we see is a lot of studies and papers coming out of people using different types of hybrids. So I've got here a study on polymer hybrids, and I've got a study on exosome hybrids as well. So this was a really nice collaboration that we did with a group at Imperial College that did a polymer-based hybrid particles for malaria disease. So it's really to interact with this blood-borne parasite. So we have some standard techniques here, as your DLS, for instance, and some small angle scattering and cryo-TEM. But whilst these are very quick, well, not cryo-TEM, but DLS at least, to analyze like, okay, do you have a reasonable population of nanoparticles? You can't really look at the hybridization from here. Sometimes you can get an indication of that from the cryo-TEM, but it's quite difficult. But of course, from a compositional perspective, we can see this quite clearly. So we do the Raman spectroscopy with SPARTA, and we can see here that when we have, for instance, polyethylene glycol incorporation, we can see that signal come up. And when we have the polymer incorporation, we can see that also very clearly. And crucially, we can determine from the Raman signal that most of these particles are actually hybrids. So we don't just have polymer and lipid-based particles separate in the solution, but we actually have combined hybrid particles that are all relatively homogeneous in composition. And we can look at the variation within those particles as well. And we can, crucially here, what they've used it for is then to really optimize their formulation. So they go to different steps and see like how much polymer could we actually add to make this formulation effective, because the polymer was really the part here that can interact with a certain charge-based interaction with this parasite. So they wanted quite a lot of that polymer. And what's crucial here, and this is something that looks a bit familiar maybe than the targeting ligands plot that I've showed you, and we've seen this quite a lot, is that you can push it to a certain amount, and then you get relatively little gain in the mean, but you can see that the variation between these particles becomes huge. And this is really what we saw here with this excessive amount of polymer as well. So it allowed them to really select with an informed way of how much of this ligand they should be adding without doing necessarily endless cell studies to verify a certain effect. And basically say like, well, these two give me roughly the same amount of ligand. Let's use the lower one because it uses less material, or what your decision might be on. Can really be based on a fundamental insight into your formulation, what is better. Right, and then let's have a look at the EV ones as well. So we work with AstraZeneca. They bought a machine about a year ago. They've been very productive in Gothenburg. So they've already published 3 papers on this. And they do a lot of work on LNP EV hybrid nanoparticles as well. Purely from having the benefits of the loading, for instance, of the LNP, and then the benefits of the targeting ligands from an exosome perspective. And there's lots of different methods that you can use to load these EVs and to hybridize them. The standard methods really didn't show them much about the degree of hybridization or how these particles are actually interacting with each other. It was just some side indications. And with SPARTA, they did a very large panel of compositional profiling here on different types of EV and LNP components to really see what this hybridization looks like. And I've got a little excerpt here, a little bit bigger. So we can see here, for instance, looking at different proteins or lipid profiles, that when we measure the EVs or the LNPs and then different hybridization, we can see that these signals start fusing together, but are not necessarily creating one homogeneous hybrid. There's still quite a bit of spread there. And this is indeed information that was very crucial for them to have for developing this types of formulations further. And we can really look at different chemical components here in much, much more detail than you could ever do with a fluorescent-based fusion assay. Great. So then I think the last part of the presentation, I just wanted to show you a little bit more on other applications beyond necessarily purely therapeutics. This was all based on therapeutics. We also work with a bunch of groups on different disease states, especially exosomes are very popular for investigating as cancer biomarkers or other kind of disease biomarkers. So this was a study where we isolated EVs from a bunch of different cells from breast cancer and healthy breast epithelial cells, and then measured these Raman fingerprints, not necessarily to look at very specific individual peaks, but more take this as a fingerprint spectrum and do it as almost like a proteomics or lipidomics based analysis, where we take all that very rich data into a multivariate analysis model which is this PCA type model that you can see on the right, to see if purely based on this fingerprinting, can we distinguish accurately between cancer cell derived or healthy cell derived particles, which we can very cleanly between there. And then we can investigate, of course, from that separation, what is it really, what causes the variation here. And we could see that the breast cancer based cells were much more lipid enriched than the healthy cells, as one of the key takeaways. And we've done a recent study here as well with the University in Canada, which was really interested in looking at the hematopoietic system and different locations in the body where EVs might come. Of course, there's a large amount of EVs in the bloodstream, but they were wondering if there's specific other nano niches basically in your body where the EV population could be conserved differently. So they isolated EVs from different locations of the body, and we've measured them here on the SPARTA as well. And it was very interesting to see that obviously when you start working with human samples, the variation goes up quite a lot. And we could see variation between the different subjects that they used, the different healthy volunteers, but we could see that the variation between the location systems was conserved. So there was more variation between locations than between people, even though there was variation within the people, if you get what I mean. So they really could show that there are these conserved different locations in the body that have different EV profiles. And again, be able to show that in a completely label-free way was a very novel application of the technology. So we're coming to the end here. I hope I didn't overload you too much with all the different possibilities, but usually for different institutes, I like to show the wide breadth of the applications that there are, because each and every one of you will work on slightly something different and wants to get something different out of it. And this is exactly a technology that does very well in a environment that has a lot of different use, because there's different strength of the technology that we can pull out. So we can look at lots of different composition, functionalization, loading, and this all leads to a better understanding and really this faster iteration cycles of optimization of your formulation to create the best formulations possible. So thank you very much for that. This is the machine that's there. So it's about this big. That you have an indication. It's about 60 by 50 centimeters, a benchtop analyzer. And we make this in London. So they're actually finished in our lab. And we work with a bunch of key partners to source, of course, the critical components inside the machine. And we provide this instrument with consumables, with software. We specifically work a lot on the software. We have excellent software engineers in-house. And a critical part here is that we take a lot of feedback from our clients to make improvements to the software. So we usually release about 2 different versions, new versions a year on the software. And this always includes a bunch of updates from what people want. Sometimes that's easy. Some people will say, like, we want more colors for the plots. Okay, that's fine. That takes 10 minutes to do. That's not a problem. And sometimes people say, can you change the whole saving structure of the system because it's confusing. So we've done that as well. But key parts here is really where we've got this Raman data, and you can get quick insights very quickly. But as with any technique, if you keep using it, you can get more and more out of it. And we want to make that part easy for you to get those deep understandings in a very quick way, without necessarily studying to become an expert in lasers or Raman spectroscopy. So thank you very much for that. That was my presentation. I take any questions you might have.
Thank you very much, Jelle, for this interesting talk and for sharing this overview of the SPARTA, how SPARTA is advancing the nanoparticle characterization. So now, if you have, are there questions?
Interesting talk. Thanks. I have a question about a technical question. The laser that causes the optical confinement is not the same that you use for Raman?
The laser is the same. Yeah,
it's the same.
Yeah. We use a one laser system. So it's not specifically tweezers, because with tweezers, they use or they use optical tweezers to move things around. We don't care about moving it around. We just want to hold it for a few seconds to take a measurement. So it's the same laser, yeah.
And then you pause, or you how you
we
release it?
use a shutter system to block the light. So yeah, maybe it's a good if I briefly explain. So what happens is you have all these particles diffusing around. We enable the light. There was diagrams here. People thought it was going a bit too deep to show the whole algorithm. We shoot the laser down, and then a particle, we optimize the concentration that a particle is relatively close there. So within 1 or 2 seconds, a particle will get pulled into the optical trap. And then we do short acquisitions with the system. It's all fully automated in the background. We do short acquisitions to see if a particle is there, because if there's no particle there, we just get water or your buffer. As soon as a particle is there, the signal goes up and we can see that there's a particle there. It's automatically being detected. And then we do a longer acquisition. You can set this in the software. It's usually a few seconds per particle to get a nice signal to noise ratio. And then once that's completed, we block the light. That particle just diffuses away. And then we open it again after a few seconds. And because these particles are diffusing in 3D, the chance that that particle diffuses in a circle back to the exact same femtoliter that we're measuring in is, of course, negligible.
Thank you.
Yeah.
Hi, thank you very much. It was really interesting. I was wondering, maybe you said, I don't know, but you said that you can correlate the size with the specific composition of a particle. How do you get information of size if you have a particle that is trapped?
Yeah, exactly. So we can do this in 2 different ways. The most common way is that if you have that particle, if it's smaller or bigger, it has more material, which will increase the signal. So the signal of the Raman spectrum is dependent on a lot of instrument parameters, of course, like laser power and things like that. That's all stays constant, of course, during your measurement. And then the other 2 factors are the amount of that material in there. So if a particle is bigger, it will have more material. So that makes the signal go up. And then specifically for different components, it depends on how strongly that specific component scatters in there. So when we look at the Raman signal, the raw signal that comes out, we can look at this in a non-normalized way. So basically raw signal intensity, and the variation there is indicative of the size of the particle. So what we tend to do is then take there was a really big peak you can saw in the middle, which is a carbon-hydrogen vibration, which is everywhere, of course, in your particle. So that's indicative of the size of the particle. Looking in a non-normalized spectra, so the raw intensity changes, we can see how variable it is between particles, and that's quite accurate. If you want to do like more absolute sizing, you can. We've done this in a paper before. You can look at displacement of an internal standard basically to calibrate that. Whilst it's technically possible, almost no one uses SPARTA for that because everyone already has a DLS. Almost everyone has a DLS. And they already know the size distribution basically of the particles. But linking that to the composition is something that you can't really do with other techniques. And that's really what we will focus on.
Okay, yeah. Now one very other fast question is, if you want to know, it's true you don't have labels, but if you want to know, for instance, if you have 2 specific proteins on an extracellular vesicle or an exosome or whatever, for that you need those proteins, you need to measure those proteins separately to check if you have them, right? Or
yeah, so we don't have the resolution to look at specific proteins on an EV, right? So it's just protein content basically and lipid content of the particle.
So you cannot say I have these 2 markers traveling within the same particle.
Not on an EV if it's just 2 different proteins. No, okay. we cannot see that. Yeah, there's different ways we could enhance certain signal of the different particles, and it's something that we're working on this year. But it's not something we can see. If we do on an LNP, for instance, people tend to put one targeting ligand on like a protein, and since the native LNP doesn't have protein, we can just look at the protein signal. Of course, that's on. But it's very interesting, especially for the cancer biomarker work with EVs, because people tend to be very focused in breast cancer specifically about HER2 protein expression, for instance. But what we can see here is that regardless of seeing HER2 specifically or not, there's massive protein lipid changes on these particles in this disease state, which is not linked to a single protein being overexpressed. So it's very complementary in that way. Mm-hmm. Yeah.
Thank you. In one of your first examples, you were
imaging the peptide
Mhm.
on the particles. I'm not sure if I understood right. You can distinguish the one that is bound to the particle that the one that is free?
So the main one that we can see is the one that's bound. Because if there's a small amount free floating in solution, because it's very confocal, we can't see the free floating one. So we only focus on what's on the particle. But if you had particles and you added some targeting ligand free in solution that wasn't bound to your particles, it would just show that there was no targeting ligand. So it's not getting confused by the fact that there is stuff in solution, but it can't separate out those two
So
signals.
you see anything that is linked to your particles.
Yeah, that's really
whatever
affecting the
leaches out, you don't see it.
Yeah, well, you can see the decrease on the particle, for instance.
Yeah,
So we've done studies like that before with drugs, for instance, leaching out of the particles, and we can see the decrease. The advantage of that is that you don't need to specifically deeply purify your sample before measuring, because we are not looking at what's free in solution until it's high concentration.
Understood now. And concerning the measurement of possible organic solvents, which is the limit of detection for that?
Depends on the organic solvent, because it depends on the specific molecule that you're looking at. For ethanol, from the top of my head, it was 0.05%, I think, in the solution. So really quite little.
And I guess the organic solvent, you can distinguish if it's one.
Which one it is. Yeah, for sure.
Thanks.
Yeah.
Okay, Jelle, thank you very much. Very interesting and unique technique. So it's very, very interesting. I guess here it's very important to the fingerprint of each component that you want to analyze, no? So if there is an overlap, I guess that this can be an issue. So is there some database already in the equipment about the different components or proteins or units that are available? Of course, then maybe it's possible to develop with new materials, but and how do you manage the convolution in case that there is a bit of overlap or not? So which is really the output of the machine? Because all these analyses are very interesting, but I'm wondering if you can get directly this analysis that you show, or how is really the way of performing an experiment with new material.
Yeah, absolutely. So when you saw the LNP spectrum, for instance, you could see these colored lines being dropped. So that's just in our software. There's a database in the software, and you can just check different components, and it will drop these lines down. And you can easily add, if you're using a novel material, you could add lines to that database as well. So what we can do with SPARTA is measure in solution other components. You could even measure dry powders if you really wanted to, as a reference sample to see where a certain new molecule might have different peaks in there. So you can add that to the database. That's very straightforward. I will say as well that there's a wealth of literature out there. The people have used Raman for a very long time, and these peaks will be in the same positions or within a few wave numbers of that position. So there's a lot of literature out there to do that. Most of the plots that you saw, apart from the 3D ones, but that's what we're adding now this year in the software, they basically roll out of the software within a few clicks. So all this univariate analysis, you just select which peak you want and press a calculate button, and it will make these different univariate plots on the surface, and you can drop in the lines to see what percentage of particles are below and above. So we do really try and make that as easy as possible.
And in terms of the sample, is there some requirement regarding the concentration of the particles, for instance, or if there are some components that emit light, for instance, and can interact with a laser, which are these in general, the requirement?
Yeah, absolutely. So so the main size that basically always works is about 40 to 400 nanometers. For some denser particles, we can go a bit smaller. If you go higher than that, you just need to make sure that they're still Brownian in motion, that they're still floating, that they don't all start to sink to the bottom. Of course, so that's a bit where the higher cutoff comes from. Concentration-wise, depends on which technique you use to measure that, but you can see that on the SPARTA as well. It gives you feedback if it's too high or too low. Roughly, we're talking about 10 to the 10 particles per milliliter. So if you measure that with NTA or so. For lipids, for lipid nanoparticles, we're looking at 0.05 milligram per milliliter total lipid content. So it usually for LNPs, I've never had a person that complained about how much that was. We usually dilute a couple hundred times or so. For EVs, it sometimes can be a bit on the higher end. And we use 100 microliters of that for our measurements. Yeah.
Okay, thank you very much.
And it's broadly compatible with a bunch of different buffers and so on. And you can measure, like a UV vis, basically, you measure a blank buffer beforehand as well. Yeah.
Thank you.
Of course.
Maybe, oh, thank you for the presentation. It was very interesting. Can you use it as well to see if you have something on top of the membrane instead of inside the membrane? Like, do you see these kind of differences as well, or maybe?
I think so. In principle, the answer is no, because if we have the Raman spectrum, we get a spectrum from a particle, right? So the whole particle. So it's not just the surface of the particle. Or, however, it's always a but. If you have, for instance, a structured membrane, like a liposomal membrane or so on, and you're putting hydrophobic stuff inside that membrane, it causes disorder in that membrane, and you can see that. You can see the disorder of the lipids inside. So in specific cases, you can. But at the basic level, in principle, we get a spectrum from the whole particle, so it doesn't distinguish between outside and inside.
Thank you.
Yeah.
I think we have from the participants online side.
Yeah.
How do we see here? Yeah.
The hybrid term in the context of hybrid nanoparticles. Yeah, so basically what hybrid means here is that you try and fuse two particles together. So if we have LNPs and EVs, what they're trying is to create a hybrid, a mixed particle that contains part of an LNP and part of an EV. And that would give benefits because the LNP might be already loaded with something, and the EV then brings in extra proteins on the surface. So rather than trying to engineer individual proteins, you try and mash these natural particles together, but quite challenging to actually know that you made hybrids. Okay, let's see. Very interesting equipment. I might have missed it, but it's not clear what is the heterogeneity of the solution that SPARTA can measure. What is the solution? Okay, I'm not entirely clear on the first part of that question. This is quite broad, but basically the heterogeneity that we measure is the difference between individual particles, right, in the solution. So if they all look the same, if they all have exactly the same amount of specific components, or if that's variable. We do, in principle, offer service as well to people that they can send samples to us to measurement, or at least please do reach out, and there's always a conversation to be had in what we can do together. Usually we try and find a way to get people at least some data, of course, from their particles to see if it works, and then we can take the discussion further towards ground applications and things like that. Yeah. Was there another one at the end? Or maybe another one? If you scroll down. So organic coating of metallic nanoparticles, yes. So if it's a pure metal, so like a gold nanoparticle, that wouldn't work on the SPARTA because it doesn't interact with the laser optical trap in the same way. However, if you have organic coating on top, like a polymer shell or things like that, that should work on the SPARTA as well. So it will then based on that organic surface, and you should be able to characterize that as well. So it's certainly worth a try to do. Yeah. Okay, great.
Okay, so I have just a final question. What do you consider the most important technical challenge that still remains to address for wider the application of the
within our technology, not in the field, you mean? Or...
No, well, maybe you can also extrapolate.
Oh, right, okay.
I mean, with the SPARTA.
With the SPARTA, yeah. So, well, I mean, I think a key thing for us, what we really like to work on, specifically for industry applications, is to get basically, we call that de-Ramanizing the SPARTA almost, in terms of like, people will want to get a result without going through the data analysis, basically. So it needs to kind of output a number or a set of numbers, a quality parameter straight away. And then if you want to know more, then people can dive into the Raman spectra. So we try and make that process as easy as possible so that people are not like sit there with a pile of Raman data and then what's next, right? So we really, in the analysis that we already got and what we're working on in the future, try and make it as easy as possible for the user to get the crucial insights that they need for their formulation. So that's a challenge because that's a benefit, of course, of if you do dynamic light scattering for size, yeah, that's basically 1 or 2 numbers that comes out and that's it on the data, right? It's also very limited in what you can then know about it, but that makes it very easy. The challenge here with Raman is for every particle, we get 2,000 data points. So we need to, it's great because there's a lot of insights, but you do need to find, your answers from that data. So that's really something that we try and make as easy as possible. I would say that's a challenge sometimes.
Okay, thank you.
Great.
Thank you very much.
Yeah. Oh, it might be one more.
Yes.
Okay. Great. Yeah. So I mean, I don't like to just throw out a number necessarily, but it is of course not the same price bracket as a DLS because it can do so much more. But it's also not the price of an MS system that is 500,000. So it's in between those two, in between these two numbers. Yeah. Well, I'm very happy to have these discussions, not on a webinar together. Yeah.
If you, well,
Of course,
after-
yeah.
Yeah, of course, they can also.
Yeah, contact us and we can talk about it. Sure.
Okay.
Yeah.
Okay, so maybe it's time to close the seminar. So thank you very much for joining me.